Dr. Gillard here. Let's start a week 2 lecture. This is the only second time I've given this lecture, so this one's still evolving. It's a tough one. It's on oogenesis, and the authors are just all over the place on this subject. So I did my best. I read all the 3-4 embryology books, histology books, to try to put this story together. So anyway, here we go. It's a tough one. So making the oocyte, the object of us here, we're trying to make a human, right, in embryology. So we made the sperm last week in week 1, but now we need to make the egg, and that is a heck of a lot more difficult than making the sperm. So let's get through it. So oogenesis, the baby is very confused. That's a really confused looking little baby. Love to see what this baby looks like when he grows up. Anyway, great author confusion. Did the best I could with this. This is really tough. And I assume, again, like I said last week, I assume you know about meiosis and mitosis and that stuff, right? I think you have that in junior high nowadays. So I assume you know that. So oogenesis is the process of making the oocyte, which is the female gamete. All this action takes place in something called the ovary. There's the ovary right there. So this whole story will be talking about how follicles form inside, we'll go this way, form inside the ovary, and they get bigger and bigger, and they finally pop and release an egg into the fallopian tube, and that's what we're going after here. Right, so it all starts now, right off the bat it's confusing because I have to jump all the way to the end of the class to talk about how this starts. So this is week 4. We get a folding of the little zygote here of the embryo. And you probably don't even know what I'm talking about yet, so you're at a disadvantage. But it's really weird where these egg cells come from. There's a yolk sac that is, we really don't use that much in humans. In animals, of course, there's yolk in the yolk sac, and it nourishes the growing embryo. But in humans, we don't do that much with it, but there is one use. It's like a highway for these little baby oocytes, and they're called primordial, means before the beginning of time. The very first oocyte cells are called primordial germ cells. And those things migrate out of the yolk sac, and they go right up the yolk sac, and then they get in to this region of the gut tube, kind of toward the caudal portion of it. And they start to hang out and build up here, and they build up and build up. So those are the primordial germ cells, but it's weird. They also induce some of the tissue around them to change, and they're going to be kind of like helper cells, and we'll see how they're going to end up surrounding these primordial germ cells. And as they get bigger and bigger, a lump right here occurs, and that's actually got a name. That's called the genital ridge or gonadal ridge, and that's made of many primordial germ cells. Okay, the interesting thing about at this point in development, about week 4 here, these could go either way. These are pure stem cells. They could go into testes or into ovaries, and we won't get into the molecular biology of how they go down the oocyte pathways. Did that say ovary? That's not right. I'll have to fix that. They could develop into the that is right. They could develop into ovaries or testes, as well as the germ cells. So could go either way, but we won't worry about the molecular biology of that. So meet the Oogonia. We have a new word. So these primordial germ cells, and there's many of them, they're dividing through mitosis. All right, so remember, is mitosis, is that a reduction division? What happens when you split into 2 cells here? Well, nothing. It's not a reduction. There's still 2N. Each one of those is 2N. Okay, so 46 chromosomes, but they are dividing. And eventually they get big enough where it changes names from the primitive germ cells into something called the Oogonia. Oogonia. Or you could call it the primitive egg. So the Oogonia cells are still multiplying and growing, but about the 3rd month, we stop mitosis and we begin meiosis, and that's a reduction division, right? And so we got to talk. It's kind of a weird meiosis because it doesn't go all the way through it. It starts in interphase, and what happens in interphase? What's the key of interphase? And the chromosomes are unwound. They're like, they're not those beautiful chromosomes. They're just like a hairball almost, massive stuff. But all the genetic material doubles. So that's the key thing with interphase. And they make it through and double their chromosomes. And I just threw this in here. Here's meiosis. Interphase would be right here. So it doesn't show it. But they make it through into prophase. But here's where the weirdness happens. So the first part of prophase, they start, and that's where crossover occurs. Remember, that's where mom and dad can swap their genetic material between alleles, genes that are similar. Mom has like a gene for eye color, and dad has a gene for eye color. Those are alleles. And they can swap chromosomes. They can swap genetic material between there, and that's called crossover. But for whatever reason, we don't understand. We know some of the molecular biology of it. I'll talk about it a tiny bit. But for whatever reason, right after the crossover occurs, the interphase stops. Okay, so they never get into metaphase. Prophase stops, and they never make it into metaphase. So our oocyte is arrested in meiosis, prophase 1. If you want, I think this might be too deep for boards, but prophase has different subcategory stages. So if you wanted to get technical, it's the diplotein stage of prophase, which comes right after crossover, where meiosis is arrested or stopped. Everybody good for that? So these little baby eggs are frozen. They didn't go all the way through meiosis. So that's kind of weird, right? You've probably never heard of that before. And at this point, they get a new name because they're oocytes. They started meiosis, but they didn't finish it. So they're called primary oocytes. Primary oocytes are stuck in prophase 1. That's like a good board thing right there. Got it? Right. This is Khan Academy. It's got a really good lecture on this. So if you could watch the Amoeba Sisters, Khan Academy is good. A little bit deeper than the Amoeba Sisters. But this is where we went. We went through interphase, and here's 2 chromosomes hanging out. So what's the diploid number of this little creature here? I don't think there is such a creature. But this imaginary creature, this cell only has 2 pairs of chromosomes. So its diploid number is 4, or 4N. My marker doesn't work there. And that means there's 4 chromosomes per cell. Its haploid number is 2N. So, but anyway, so that's a germ cell. Only ours is, the diploid number, of course, is 46. Haploid number is 23. But we go through interphase, and you can see that these chromosomes have doubled their DNA. They've doubled their genetic material, but they still have 1 centromere. Remember, you always count centromeres, and that gives you the chromosome count. This one has 4 chromosomes. This one has 4 chromosomes, but it has 8 chromatid, right? So there's 2 chromatid on each. These sister arms here are called chromatid, right? So interphase is done. Next, we go into prophase 1, and here's where crossover occurs. And the professor just showed an example of 2 arms touching here, and then the genetic material swapped. So you see when crossover is complete, we have a piece of mom's chromosome on dad's chromosome and a piece of dad's chromosome on mom's chromosome. And that's what crossover is about. But this is where our eggs are stopped right now. Our primary oocytes are stuck or arrested in this phase. Everybody good with that? Why does meiosis stop? We're not 100% sure. We do know that there are some chemicals, there's some biomolecules that are involved in this. So we know that the cells adjacent to the oocytes, or the Oogonia, I guess we are at this point. Well, we're in between oocytes and Oogonia. But nevertheless, adjacent cells start producing cyclic GMP and cyclic AMP. And that soaks in to the Oogonia, and that's got something to do with freezing them. How do we know that? Because later, when we're about to have ovulation, if you block the production of these things, ovulation occurs. So these are two important biomolecules that have something to do with meiotic arrest of oogenesis. Okay, got it? All right, just a note here, everybody. I don't know, do you guys still know the story of Sleeping Beauty? Sleeping Beauty, she was awoken by a kiss from her prince. But the point of this slide is that some of these primary oocytes are stuck in prophase 1 for 5 decades, right? A handful of the primary oocytes wake up each month during the female's menstrual cycle. But there's thousands of them, and most of them are asleep, and they can be asleep until it's their turn to wake up. And if menses occurs into the 40s, that's 5 decades. That's why women in their 40s, it's risky to have a baby, because they've been asleep so long. They've been exposed to teratogens and x-rays over that 40 years, and there could be chromosomal damage within these primary oocytes. Okay, that's the story there. So waits a long time. Everything I said. So the nucleus is meiotically arrested. Here's another thing that helps. Did I miss something here? Oh, yeah, there's the question. So how can they wait? How can they be preserved for so long? Because plenty of people in their 40s have successful babies with normal babies. So how can they be asleep for 5 decades? What protects them from teratogens in the environment? And so we know a little bit about this. They begin to accumulate fluid in the nucleus of these primary oocytes. And this fluid somehow kind of cushions and protects and absorbs UV radiation and things like that. It protects them from the environment. So that little fluid is really, really important for them. All right, now let's meet a follicle. Here's our first follicle. So this is a little confusing too, and the authors don't agree. So about the 4th month of development, a single new layer of simple squamous cells, and you've had histology. I think you know squamous cells means flat. The cells are flat. So we get a single flat layer that's called simple. Simple means that it's only one layer, and squamous means it's flat. So we get a flat layer, a single layer of cells around the primary oocyte. And probably derived from some of the surrounding ovarian cells. They're not 100% sure where they come from. It doesn't look like they have to come from the environment somewhere. But nevertheless, we get a ring around the primary oocyte. And that ring has a basal lamina on it as well. And these are called follicular cells. Right, so they get a new name. This layer of simple squamous cells is called follicular cells. And you could say that this is a follicular epithelium. So Carlson also calls them granulosa cells, and some of the other authors save that word until they become multi-layered. But you might hear granulosa cells. But if they said granulosa cells, they would have to say there's a single layer around the primary oocyte. And so, the primary oocyte could look like this. It's just we have a ring of flat cells around them. Simple as that. And those our follicular cells, and now we have a follicular epithelium, complete with a basement membrane or basal lamina. So now we have a creature, we have a primary oocyte with a layer of epithelium around it. So that whole structure gets a new name, and that's called the primordial follicle, not the primary follicle. Don't confuse those, because primary follicle is coming next, and then there's a secondary follicle, and even a tertiary. Or we'll get to that when we come. But right now, this is primordial. Okay, what's a primordial follicle? It's a combination of the primary oocyte with a single layer of cells, specifically a follicular epithelium around the primary oocyte. That's a primary follicle. Are we good with that? By the 5th month, there are about 7 million primordial follicles in our ovary. Those cells have been dividing, other cells have been dividing to create an ovary. And this is going on within the ovary. So our primordial follicles are within the ovary. And by month 5, there's 7 million of them. That's like crazy. And then all of a sudden they start dying. And we don't know that's normal, but we don't really know why that occurs. But a lot of them die. In fact, by the time we're in month 5, by the time you get to month 9 and you're born, you lose way over half. You're down to only 2 million. So you've lost the majority of them just through development, and we don't quite understand what the deal is with that. And then even more of them die. They don't do very much after the child is born until the child starts kind of secreting hormones around 12 or so. But by the time puberty begins and secreting hormones and you start to become a woman, which is called, by the way, menarche. So at the time of menarche, we don't even have 1 million left. We started with 7 million to 2 million at birth, and now we're down to 40,000 of them. So, and to go a step further, we're going to learn that some of these primary oocytes or primordial follicles, they wake up and they start growing like crazy. And we're not sure why some wake up and others don't. But a whole bunch of them will stay in that, just a primordial follicle. But only 400 of them will ever even wake up to kind of play the great race game to see if they can create a really oocyte, a secondary oocyte that has a chance to become fertilized. Carlson says 99% of germ cells are lost via apoptosis during woman's lifetime. Most of this occurs prenatally. So about 12 years, all of these 40,000 primordial follicles, they lie asleep. Nothing happens. However, when menarche occurs, when some FSH and LH, luteinizing hormone, follicle stimulating hormones, start flowing in the 12-year-old, they start getting cranky and start turning into a young woman and have their period, some of those wake up. And then the great race begins, and it is a great race. So it's a race to see who gets the rare chance to actually escape the ovarian prison. These 40,000 primordial follicles are trapped in the ovary. Most of them will never get out. They'll live and die there. But a few of them get to escape, and that's the great race. So what wakes up the primordial follicles? So each month at the start of menarche, because girls have a cycle every month, right? Anywhere from 5 primordial follicles to 50 suddenly wake up, and they start growing. They start growing in size, the follicular cells, and we'll look at. The oocyte doesn't grow, though. The actual thing that's going to be the egg does not grow, so it's important to understand that. It's the cells around, the follicular cells around them that start growing, because right now we have a single layer. Pretty soon it's going to be a double, then a triple, and it's going to get bigger and bigger. But yeah, so they start growing to see who can grow the biggest and strongest, and biggest and strongest will rupture out of the ovary and get into the fallopian tube and have a chance to be met by a sperm. And the authors, what really wakes up, the authors are all over the place, but there are some hormones that are the key for development. We're not sure what initiates the thing to wake up, though. Of this group, only 3 of them will reach. So of this 5 to 50 primordial cells, or primary follicles that wake up and start growing, 3 of them will take the lead, and they'll eventually destroy the rest of them. So only 3 of them will have a chance to finish the race. Okay, what about some hormones? So primordial follicles that wake up each month need hormones to drive their growth. Later on, that's not true, but when they first waking up, it's a hormone-dependent event. Remember, when a girl starts her period, the pituitary releases FSH and LH. Who signals the pituitary? Something called gonadotropin-releasing hormone. So the clock is in the hypothalamus. What tells the hypothalamus it's time to make the female go through a menstrual cycle? That we don't understand completely. But the key is that luteinizing hormone, follicle stimulating hormone, will hit the ovary, and why all those 50 follicles or 4 to 50 follicles will start growing. Why the other ones don't grow is a mystery. But maybe next month some of them will grow. So there's a lot we don't understand about this. All right, so hypothalamus releases gonadotropin-releasing hormone from the anterior pituitary, and it stimulates the anterior pituitary to release what are called collectively the gonadotropes. You should know this word. Probably won't have time to cover this when we get to 5th quarter. We'll do quite a bit of endocrinology, but this we haven't gotten to yet. There's never enough time. So many diseases. Follicle stimulator hormone is the main one that drives this follicle development. Folliculogenesis, it's sometimes called. And we could get crazy complicated, right? But we won't. Inhibin is the thing that does shut things down. We'll talk about this, I think, the very last slide. But the follicle, what's left of the follicle, actually secretes inhibin and shuts this whole mechanism down, as we'll see. So gonadotropin-releasing hormone, some fun facts about this. This is a very strange development of GnRH, is really strange. So the neurons that spit out and make gonadotropin-releasing hormone, they're in the thalamus, or the hypothalamus, just like the let's see, what's an example? Antidiuretic hormone. That's made in the hypothalamus and transmitted down into the posterior pituitary. So these are also made. Gonadotropin-releasing hormone is made there. But the point is, this is really weird. Embryologically, the neurons that are created are not created in the hypothalamus, which is really, really strange. Come to find out, they're actually created on something called the nasoplacentode, which is going to be the nose holes, the choana, and the nares. So that'll form into the nose. So it's really weird. And the nerves, the GnRH neurons, are actually born here, and then they migrate across the surface of the brain to their location in the hypothalamus, and then they finish their differentiation there. So really weird. But that's a long migration, and things can go wrong with it. And there is a syndrome called Kelman syndrome, where the migration never happens, and the child can't go into puberty because of that. Pretty rare, so I'm not going to get into that any more than just mention its name. Gonadotropin-releasing hormone receptors they live in the anterior pituitary, of course, and they're stimulated by the when they're stimulated by GnRH, they cause the anterior pituitary to release the gonadotropins, which is luteinizing hormone and follicle stimulating hormone. Okay. All right, so we said it's a race, so let's talk about the race. So in order to grow, you have to be able to soak up follicle stimulating hormone. So the follicle cells, the cells around our oocyte, they have FSH receptors. So FSH can bind to them and stimulate them to grow. And it's a cascading system where one product goes into the nucleus and turns on the genes for things, but we won't get into that deep here. So just know that the follicular cells do have receptors for FSH, and when FSH is a hormone, when it binds, it causes the growth to occur. It also kind of stimulates them to produce a small amount of estrogen, which turns around and helps them to grow as well. Okay, so now we're going to say goodbye to the primordial follicles, and we're going to meet the primary follicle. Don't get those two words mixed up. So the first thing that happens is a cell type change. So now this is controversial, but when do you have primordial follicle versus a primary follicle? What's the change that has to occur to create a primary follicle from a primordial follicle? Some authors say that the first thing that happens is the squamous cell type. Remember, we have a simple squamous that surrounds the oocyte or the follicular epithelium. It morphs into a new cell type. In fact, it becomes cuboidal. And that's in response to FSH. The simple squamous cells, they morph and thicken into a cube-like structure. Some authors say that they're more columnar-like. But nevertheless, once that change occurs, some authors say you don't have a primordial follicle anymore. Now there's evidence of growth. This is a primary follicle. Other authors say no, it's when you have more than one layer. That's when the primary follicle is born. So there's kind of a split decision, and two board books, both one says one thing, one says the other. So hopefully they'll stay away from that question. So again, arguably some authors say that once the histological change occurs, primordial follicles morph into primary follicles, aka primary growing follicle is another word for it. Okay, and don't forget now the primary follicle. Nothing's happened to the primary oocyte, which is still stuck in prophase 1. That's not going to change for a long time. We're going to get some growth out of this thing. And then the darn thing's going to get stuck again. But right now, it's still that primary oocyte. These are the cell layers going on outside, wrapping around this, is that we're talking about. All right, so now we can have our first picture of a primary growing follicle or primary follicle. And there's our oocyte, which is stuck in prophase 1 here. It's not shown. But we have a nice layer of cells around it. They're cuboidal-like, right? They're not flat cells anymore. They're cuboidal, so therefore this is called a primary follicle and not a primordial follicle. Are we good with that? All right, and we can still see we have a basement membrane here, basal lamina, which is on the outside. We're going to grow something really important we've talked about last week, didn't we? The zona pellucida. We talked about that last week, right? We're going to grow that thing next. So let's meet it. Let's do it. So the racing primary follicle, primary growing follicle, now develops a new layer to separate it from those cuboidal cells, and that's called the zona pellucida. Simple as that. We'll leave it at that. The oocyte and the follicular cells make up the primary follicle, as we said. So they, specific to the zona pellucida, there's 3 different genes that get turned on that are important. And they secrete 3 proteins which help make that zona pellucida. So we have zona pellucida protein 1, 2, and 3, or we can call them ZP1, 2, and 3. That sound familiar, ZP3? Because we talked about that last week. The other gene products we won't get into. There's other proteins that are used to make zona pellucida as well. But the big one is ZP3, because that is the binding site for the sperm head, right? So hopefully the gene for ZP3 isn't mutated, or we're going to have a problem. ZP2 actually might have a binding site for sperm. It's like a secondary binding site as well. All right, so back to the future. If we go back, way back to last week, remember that we had a SED1 protein here, and it's binding to this blue ZP3 protein. And there's the GALT-T. That's another double binding system there as well. Communication. So now our oocyte is trapped inside the zona pellucida. Is there any way to communicate with the inside of that thing? And there is. So the cells that are around the outside are able to communicate through the zona pellucida by two types of projections that penetrate the zona pellucida and sink into the oocyte. Specifically, we have a ATZP, a protein, which connects to the plasma membrane of the oocyte. Plasma membrane, I didn't say this, but there's actually microvilli, little hairs that come off the oocyte plasma membrane, just like the enterocytes have microvilli. So do these. And these AZPs connect to that, and via gap junctions, messages can be sent from that outside layer into the oocyte itself. MTZP also hooks up with the oocyte, and paracrine signals are like hormones that don't use the blood, but they use the kind of the interstitium to swim. So those are two ways that the outside world, like follicular cells, can communicate with the oocyte. Folliculogenesis is a new word. Larsen's the only one who used that. It's not a board book, but it's a good word because we're developing a follicle, right? The oocyte's not doing anything. It's just sitting inside the follicle. So oogenesis is the overall process of making a secondary oocyte. But really the process of getting the oocyte out of the ovary is called folliculogenesis. Right? So after the zona pellucida forms, we've done that. What happens next? Well, we have a single layer of follicular cells. They're going to start multiplying through mitosis. And it's going to become a double layer and then a triple layer. So that layer outside the oocyte is going to start to thicken. And that's folliculogenesis. And yep, so our primary growing follicle, sometimes it's just called the growing follicle, but should call it primary because there's a secondary growing follicle as well. Okay, and remember, while this is happening, not all of the primary oocytes are undergoing this process. The rest of them are all suspended with a single layer of follicular cells around them. Right? They're primordial follicle. Those are not only about 50, probably about 25 of them have been selected and are growing at this time. The rest of them are still kind of in suspended animation. Ross is a really good medical histology book. So he points out a couple things. There are 3 types of follicles, ovarian follicles. There's the primordial follicle. Those are the ones that are sleeping, waiting for their chance to wake up and race. There's the growing follicle, and that's where we are at the story right now. Specifically, we're at the primary growing follicle stage. And then there's the mature follicle. Sometimes it's called a tertiary growing follicle. Most of the time it's called a graafian follicle. That's where we're headed. Okay, so that's kind of where we're going. This thing is going to get bigger and bigger and bigger. So let's turn to our story. We're still at the primary growing follicle stage. So at this point, we do have multiple layers. Now the follicular layer has got 2 layers or 3 layers maybe at this point. And once it has 2 or 3 layers, you don't call it the follicular layer anymore. Now you call it the granulosa, stratum granulosa, or membrana granulosa. Okay, but it's just multiple follicular cells that have grown. But you don't call them follicular cells. You only use the word follicular layer, follicular cells, when there's a single layer. Once it becomes more than 1 layer thick, you use the term follicular cells or granulosa cells. Okay? Some authors use them as AKAs. All right, now some more. Everybody know who that is? The original Mr. Spock, one of the greatest TV series of all time, in my opinion. Even though the video and the technical stuff we can do today, it was really basic back then, but the stories were still really good, I think. Anyway, Ross says the zona pellucida forms. Yeah, this is a little problem. So Ross says the zona pellucida forms after the follicular layers start proliferating into multiple layers. But Larsen says, nope, zona pellucida, the zona pellucida has already formed before proliferation start. So that's just an example of how authors say different things. But nevertheless, we've talked about the zona pellucida. Our follicular cells have turned into granulosa cells because they're multiple layers. Let's take a look at what we have now. There's our lazy primary oocyte still stuck in prophase 1. It's got a zona pellucida around, but now it's got way more layers. Look at all these layers of cells. So we don't call these follicular cells anymore. These are granulosa cells, and this is the granulosa layer or stratum granulosum. Okay, the basement membrane is also still there. It's just been pushed out. I think, did I have a mistake on the slide? Once the granulosa cell layer is thick enough, a basement membrane forms. This basement membrane has been there. I'll try to remember to change this. That basement membrane has been there since a long time ago, so it doesn't form now. It's just been pushed out. But there is a new layer that is going to form. Oh, maybe is this diagram the one that's warm? No, there's the basement membrane. No, we're good. So there's a new layer that's going to start forming now. These granulosa cells have formed inside the basement membrane, but now we're going to have some cells start popping up outside the basement membrane, out toward the periphery. And this new layer of cells has a new name. It's called the theca folliculi. Theca folliculi. Make sure you know that. There it is right there. Theca folliculi. Consists of 2 layers. There's the theca interna, which is a lot of blood vessels. The cells in there secrete angiogenesis factors. And it's got collagen and fibroblasts in there as well. But these cells have the ability to produce steroids, as well as they have receptors for luteinizing hormone. The theca externa is the external part. This is really important. These cells have created smooth muscle, kind of like a tunica media. And so when hatching occurs or ovulation occurs, the winner, the follicle that has grown the biggest, it'll contract these smooth muscle fibers to have the oocyte squirted out. So we got to have smooth muscle fibers in there as well. Okay, now here's another weird thing that happens. So when you get when this stratum granulosum is 12 cells thick, you start forming little cavities here and there. And then the cavities start hooking together to make a bigger cavity. And that's called the antrum. The antrum. Okay, so once the antrum forms, we get another name change. So histologists and microbiologists and such, under the microscope, once you start seeing an antrum, you don't call it a primary follicle anymore. Now it's a secondary follicle or secondary growing follicle. Some call it an antral follicle, but secondary or secondary growing follicle is what I'll call it. And guess what? That thing is going to keep right on growing. Okay, we're going to talk about a chorionic cavity. That's the same sort of thing when we talk about a zygote in an embryo. But same thing. It's going to eventually, this cavity is going to eventually split this stratum granulosum into 2 layers. But it's not going to split a stalk of cells, which is exactly what happens with our bilaminar disc. So we're going to get a stalk right here in a second. Getting ahead of my slides. That antrum is filled with a fluid and a lot of hyaluronic acid, which is a glycosaminoglycan, which we'll talk about in spinal anatomy. And the fluid is typically called a follicular fluid or a liquor folliculi. As the secondary follicle grows, so the antrum is going to grow, just like I said. Primary oocyte, still stuck in prophase, nothing's happened there. But that little stalk I talked about is going to be called a hillock. A hillock. Carlson says that when the antrum that grows is going to split, it's going to split, where is it? It's going to split the stratum granulosum into 2 layers. And so let's look at that. The 2 layers would be an outer layer called the mural granulosa cell layer, and the inner layer would be called the cumulus orpharus layer. Crazy words, right? Got to know those words. Okay, so in this, now look at how we've progressed. We have a gigantic antrum here, and we've split our stratum granulosum into 2 layers. We have a mural granulosa here, and we have a cumulus orpharus layer here. Does that look familiar? That oocyte? Zona pellucida? That's the corona radiata. This is going to be ejected with the winning oocyte. So that's going to turn into the corona radiata. Okay, I think I got it there. So the stalk region is also, some authors call it the cumulus orpharus as well. All right. Okay, so out of the 50 or 25, or how many converted into primary follicles and started the race? By this point, once this is created, 3 of these happen about the same time, and the other ones are lagging behind. And so the ones that lag behind are not going to make it. So 3 will be left, and typically only 1 will develop. The most developed one is the one that gets to ovulate. The other ones are destroyed. They just kind of degenerate away. How do the big 3 survive? How do they kill off the other ones? Well, the big 3 are ahead in development, and they actually become FSH independent because they're going to secrete their own FSH. And when they don't need FSH anymore, they also secrete something called inhibin, which I showed you. Inhibin is released, and it turns off the FSH and LH secretion, for that matter. So the younger ones that are still FSH dependent, they'll die without FSH. The bigger ones don't need FSH anymore. Did they secrete their own? I don't think that's true. They just don't need FSH anymore. They don't make their own FSH. Okay, start the 24-hour clock. So in 24 hours, we're going to have a winner of this race, and we're going to have an ovulation. So the largest follicle, once it reaches the diameter, the magic 10 of about 10 millimeters, it changes names again. It's not a primary, it's not a secondary follicle, not a primary follicle. It's called a graafian follicle. Some authors call it a mature follicle. The board books are not going to call it a mature follicle. That's too easy. They're going to call it a graafian follicle. Some authors call it a tertiary follicle, which is nice, but I think it's going to be too easy. I'm pretty sure the boards would use graafian follicle. This follicle will be around for about 24 hours, and then it's going to be destroyed. But the oocyte is going to be catapulted out of it. Well, not catapulted, but it's going to get shot out of there. How does it get released? So here's the final day. So about day 14, that's the ovulation day, right? In the morning of day 14, there is a huge surge of gonadotrophs. FSH and LH are released from the pituitary by mechanisms we won't worry about. The surge stimulates the follicle's primary oocyte to resume meiosis 1. What? Yes. Sleeping Beauty, our primary oocyte, we really haven't talked about it much, it's finally going to do something. So with this big surge, we are going to get the stimulation. Specifically, it's luteinizing hormone, which gets in. It actually shuts off the cyclic AMP, which was kind of suspending it and with fluid anyway. But it's going to wake up and start to go. It's in prophase 1. So what's IP mat, interphase, prophase, metaphase. It's going to go through metaphase, anaphase, telophase, cytokinesis. Yeah, so it's off to the races again. Also, the cumulus cells, remember, they're the ones that have the luteinizing hormone receptors. So those cumulus cells, once they hook up with luteinizing hormone, it shuts down those gap junctions. And so now you can't get a flow of cyclic GMP or cyclic AMP into the oocyte anymore. Remember that cyclic AMP and GMP, especially cyclic GMP, that was the one that was kind of putting it into sleeping beauty phase, we think. It was part of it. So once those levels drop, meiosis starts up again, and prophase is off to the races. Okay, remember meiosis 1, that's a reduction division. So we're going to get two products after meiosis 1 is completed. We're going to get, a secondary oocyte, which stole all the cytoplasm. We're going to get another little body, a little oocyte kind of thing, but it doesn't have any cytoplasm because there was unequal cytokinesis. And so that it's called the first polar body, and it doesn't have enough to really stay viable. So meiosis 1 is reduction division. So we just created two cells from our primary oocyte, the secondary oocyte in the polar body, but it's a reduction division, so we just split everything. So now we have a haploid, or we have a 1n23 chromosome creature, which is haploid. Okay, the polar body too has 23 chromosomes, even though it's not really viable, but it still was a haploid reduction. So great, now we're going to start meiosis 2. Awesome, and guess what happens? We get stuck again. This time it makes it through interphase, or there's no interphase. It makes it through prophase, and then when we get into metaphase 2, it gets stuck. Just like this little koala bear, I guess there was a newspaper article, this little guy kept getting his head, how it got its head in there, and it couldn't get its head out. So he's stuck, and now we are stuck in prophase 2 of metaphase. All right. In fact, this oocyte may never get out of metaphase. It may be stuck in there forever. The only time that will turn metaphase back on and you'll actually finish meiosis is if a sperm fertilizes this oocyte. That's the only thing that can kind of get it unstuck. All right, let's hatch. So on day 14, that cumulus orifice breaks down. The hillock of the cumulus orifice, or that little stalk, will break, and the secondary oocyte is loose and floating around in the antral cavity. And over the next few hours, the remaining cumulus cells start secreting hyaluronic acid. That binds water like crazy. So this graafian follicle is going to swell up like crazy. It's going to swell up so big you can actually see it bulging out of the ovary with the naked eye. Okay, because water binding, I mean, that's what it does. Get massive expansion. The graafian follicle doubles in size to 20 millimeters. Okay, so at this point, the secondary oocyte and cumulus cell mass puts pressure on the ovary follicle and the wall, so the oocyte is starting to rupture out of the follicle that it's in. In order to hatch, the follicle wall must break, and currently we don't have enough pressure, so we have another problem to solve. It's getting big, but the wall is too strong, so an inflammation starts. The theca folliculi start releasing histamine and prostaglandins, and the inflammation starts to break down the wall. Okay, and also the graafian follicle is so darn big, it's causing a visible bump. Here's an endoscopic view or laparoscopy view of a bulging ovary, which is ovulation is about to occur. We have a secondary oocyte that's going to rip right out of this in a little while. It's almost like it's given birth, but there's no vagina. It literally rips out of the ovary. That bump has a name. That's called the stigmata, or the follicular stigmata. Okay, so theca externa, remember it has the smooth muscle, so that's going to finish off the job. So we have an inflammation that weakened all the wall. Secondary oocyte is under pressure. It's being squeezed outward. And yeah, so smooth muscle contracts and squeezes that secondary oocyte. And it also, if that wasn't enough, it also releases some collagenases, some collagen-degrading enzyme, which also breaks that follicular and ovarian wall. And now the secondary oocyte is born, and it's outside the ovary. The race is done. But now it's got to get in the fallopian tube. So down the hatch, the fimbriae of the fallopian tube will grab that little secondary oocyte and sweep it right into the lumen of the fallopian tube. Hopefully it won't get out. If it gets out, maybe it gets out here in the ovary and the sperm find it, you got yourself an ectopic pregnancy. What happens to the broken sac now? What about the follicle? The graafian follicle is like a sac of cells. We're going to use that thing. So after ovulation, those granulosa cells, they start proliferating through mitosis again, and they create a new cell type. Those are called luteal cells. And the luteal cells take over and fill up that broken sac, and now we have a healthy sac of new cells. The luteal cells collectively are called the corpus luteum. So the tertiary follicle or graafian follicle after ovulation becomes the corpus luteum. Everybody understand that? That's really important. And that's considered an endocrine structure because the corpus luteum secretes some important steroids. So specifically, let's look at what the corpus luteum does. It prepares the uterine endometrium for fertilization. It's going to assume a pregnancy occurs, and it's going to release estrogen and progesterone, and luteal progesterone will stimulate the endometrium to become thick and vascular and get ready for the kind of impregnated oocyte, which is heading its way, hopefully. If it fails, if fertilization doesn't occur, then the corpus luteum degenerates into a scarred-like tissue, and that has a name. It's called the corpus albicans. Corpus albicans. All right, so I know that was a tough lecture. That was a tough one for me to make. There's not even any good YouTube videos on that thing because it's so tough. But now you have a good understanding of it. All right, we'll see you later.